Hello, everyone. Welcome to today's live broadcast, Close Your Uncertainty Gap with High Resolution Mass Spectrometry. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by LabRoots.com, the leading social media site for science professionals, and sponsored by Thermo Fisher, a world leader in serving science whose mission is to enable customers to make the world healthier, cleaner, and safer. Thermo Fisher Scientific helps their customers accelerate life sciences research, solve complex analytical challenges, improve patient diagnostics, and increase laboratory productivity. For more information, please visit www.thermoscientific.com. Before we start, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button on the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenters. We are proud to welcome Kristine Van Natta, Strategic Marketing Applications Specialist with Thermo Fisher Scientific, and Maciej Bromirski, Senior Applications Scientist with Thermo Fisher Scientific. Our first speaker is Dr. Maciej Bromirski. He received his Master of Science in Physics from the University of Manitoba, Canada, followed by a business degree at Georgetown University in the USA. Maciej started his career at MassTech Incorporated as a research scientist working on novel TOF instrumentation. He then moved to Europe to work for Waters as a product support specialist. Upon his return to the USA, Maciej rejoined MassTech and in 5 years progressively moved to become the Director of Business Development for all AP multi-ion sources. Since mid-2010, Maciej drives the development and global marketing of the Exactive portfolio product out of Bremen, Germany. I will now turn it over to Dr. Bromirski.
Okay, thank you, Judy, for the kind introduction, and thanks to all for joining this today's webinar. So my webinar presentation will focus on closing your uncertainty gaps in forensic toxicology labs with high-resolution accurate mass instrument. So really the focus of my presentation will be on the newly introduced product, the Exactive Focus mass spectrometer, which was introduced about a year ago at SOFT conference 2014 in Grand Rapids in the US. So before I jump into the really core of my presentation, I'd like to give you a quick overview of Thermo Fisher Scientific being the world leader in servicing science. We're truly a global company with over 50,000 employees in 50 different countries with unparalleled commercial reach. We have unmatched depth in leading innovating technology, applications expertise, and lab productivity. Thermo Fisher Scientific has 5 premier brands with ranging from Thermo Scientific providing mass spectrometers, LCs, and other products to Unity Lab Services, which provides customer service for all products. So now let me start really with the core of my presentation, focusing on the Orbitrap technology and on the Exactive family product. So here in this first slide, I want to show you on the left-hand side a typical instrument that you see in the lab, which is a single quadrupole, triple quadrupole, and an ion trap. These instruments are predominantly used for confirmation and quantitation of really known analytes. The Orbitrap instrument on the right-hand side is really a new technology for your lab that not only can be used for confirmation, quantitation, but also for targeted screening, unknown screening, for identifying new substances, for identifying unknown substances as well, and having the ability to do retrospective data analysis. Okay, so I guess the key thing here is that you can screen to identify with high resolution accurate mass instrument. So why the Exactive Focus for forensic laboratory? There are really four key topics here. First one is versatility. The ability to do targeted screening, to do unknown screening, to do quantitative and confirmative analysis, as well as bulk substance analysis. The Exactive Focus provides ultimate performance in high sensitivity, high selectivity, really a true high resolution and accurate mass performance in data acquisition, and you have the data confidence in the results because of all these key aspects of the instrument. Instrument is really easy to use, has a really simple calibration, which takes about a couple minutes. There's no need for optimization, like optimization of transitions and collision energy like you do on a triple quadrupole instrument. And it's really a robust platform. And the last key feature which I really want to point out is the affordability. This product is priced competitive to high-end triple quadrupole with all the other abilities that you can do just like you do on the triple quadrupole in terms of quantitation, but you can also do screening of target and unknown compounds, and it's really a low maintenance instrument, right? So if I go to the next slide, what is the power of the Orbitrap? I want to give you a few technical performance of the instrument. So first being the resolution, up to 70,000, a massive charge ratio of 200. The instrument provides stable scan-to-scan max accuracy of less than 1 ppm, sensitivity at sub-femtogram levels with a dynamic range over 6 orders of magnitude. In addition, the instrument can run from mass range of 50 to 2,000. You can do on-the-fly corollary switching, so you can record one full cycle under 1 second. At 35,000 resolution setting, that means that you can acquire a positive and a negative scan under 1 second, which requires no recalibration of the instrument from why are you doing polarity switching. The instrument has a software interface which is modified for routine workflows, so making the instrument really simple to use, really a drag and drop method of building your work for the experiments that you're choosing to do. It's really easy to set up screening and quantitative methods, which we will see in later. And it's really robust in terms of operation, with calibration holding for a number of days. So first, let me show you schematic diagram of this instrument. So here we see on the bottom right-hand side ion source, where the ions are created, and the ions fly into the mass spectrometer, into the RF lens part of the instrument, where they are focused inside mass spectrometer towards the bent flattable. Inside the bent flattable, the ions are selected from the neutral, so that kind of helps to really create high robustness and reduce any contamination of the instruments. The ions are then guided to the quadrupole mass filter, where there is the precursor selection. So you open up your isolation, or you narrow down your isolation window, depending on the type of different experiments you want to do. Then go into the C-trap, where you store and cool the ions. And there you have actually sort of a selection. You can either put them in the HCD cell for MS fragmentation and then get them back into the Z-trap, or you can direct them into the Orbitrap on the bottom here. So you can, with the different type of experiments, you can get a full spectra, so full scan spectra. You can get a mass spectra. You can choose to just select the ions in a selective ion monitoring or SIM mode, so where you select it and then detect it, or you can do a PRM, which I will explain in a while. So let me just show you how the Orbitrap really works. So what we have here on the bottom is we have the Orbitrap, the cross-section of the Orbitrap. So you have here and here 2 outer electrodes. So we call them the outer electrode, and the one thing in the middle there, it's called the central electrode. So ions, as you can see, are illustrated in these 3 different rings of 3 different colors. They oscillate between the 2 outer electrodes along the z-axis of the central electrode. And this oscillation actually, as you can see, at different speed, it represents 3 different ions of 3 different masses. And really the quickest, the fastest oscillation, being the yellow, represents the low ion, and the slow is the green one, which is the high ion. So when these ions are oscillating back and forth, they induce a current on the outer electrode, and that current actually creates a transit, which is shown here on the top right-hand side. And that transit, if you take a Fourier transform of that and you calibrate it, you get a spectrum, and that's it's shown right there. So you have a mathematical formula where the oscillation of the frequency along the z-axis is proportional to the square root of m over z, and actually you get a spectrum on the top. So that's how the instrument works in a very quick and simplified way. So now let me show you a little bit of performance in terms of resolution, because I think that's key aspect of this instrument, right? Because the resolution is the key performance feature which allows you to get accurate mass, because when your resolution is high enough, and there are many papers stating that you need at least 50,000 mass to charge ratio of 200 to get enough resolving power to be able to separate your ions of interference from your interference. Only then you're able to get the accurate mass assignment of that compound of interest. So here's just an example of really calibration mixture, the direct infusion in positive mode of a calibration mixture. We have a few things here on mass to charge ratio around 200 or 195. We have caffeine, and it shows you here that the resolution is more than 80,000. And then we have a couple of peaks on the right-hand side. These are all polymers, and ranging from from 1222 at mass at resolution of more than 32,000 to mass to charge ratio of almost 2,000 there, with a res of 25,000. So that's what you can expect on all your samples in this mass range, right? So now, how does this compare to other competitors, which is the QTOF? You can see that the Exactive Focus resolution is totally superior to any QTOF with 3 different vendors, as I might put here: vendor S, vendor A, vendor B, and you can think those are. But if you look at the resolution, what's very important here is really the big difference, the big gaps, and the low mass range. And this is very critical for all your small molecules, because this is where your molecules are, and this is where you need the high resolving power to get accurate mass assignment of your targeted compounds or of your unknown compounds. Okay, so now I'll give you an example of resolution. Now I'll give you an example of scan-to-scan mass accuracy. That's really important, right? Because the ability to measure, your compounds while the LCU is running, scan-to-scan is very important. You don't want to sum up scans. You don't want to, be able to. You don't want really to scan on many, sum up many scans and get an average mass. You want to get scan-to-scan identification and scan-to-scan and good mass accuracy. So that's what's represented here. And as an example, we do carbyl, that's the elemental composition there, with a resolution setting of 70,000, and we have a 5-second LC base peak, so base to base about 5 seconds. We have about 80 positive scans. Now, if you look at scan-to-scan, it's less than 1 ppm as well, under 1 ppm, with an RMS value of approximately 0.62 ppm across the peak. So that's an excellent result there. So you can really be confident with your results, because scan-to-scan mass accuracy is just excellent. I don't have time to show you more, but I can mention that it's not just within those 5 seconds. I mean, this is actually, if you're doing acquisition for many injections, we had an example where you had about 1,000 injections, which took about 120 hours. You see constant mass accuracy throughout all these injections. So you can, because the instrument stays calibrated and remains in its calibration for a very, very long time. And you don't need to recalibrate from injection to injection, nothing like you have to do with other instruments. So I want to list out a couple of 10 reasons why the Exactive Focus is better than the QTOF. So let me start with the bottom. So first of all, you get a one pump, so it's very quiet. There's only one signal for pump. Orbitrap is your analyzer, but also your detector, right? So it's really a robust detector, robust analyzer, and there's no need to exchange it like MCPs and QTOFs. You have calibration, you have laboratory productivity with small footprint and really benched-up mass spectrometers because it's only about meter by meter by meter. You have experimental flexibility with easy to use, really a plug-and-play device. You have fast polarity switching, which I mentioned in the past, so you can quickly get positive and negative ions. You have mass stability and mass accuracy, so the calibration remains for days, 2 weeks time, and unlike any other instrument on the market. You have maximum scan-to-scan variation, so you have excellent signal-to-noise ratio. There's no need for averaging, and it's really HPLC compatible. You get enough scans for both screening and for quantitation when you're using or when you're running HPLC method. There's no trade-off between resolution and sensitivity at all. So there's no compromise between resolution and sensitivity while you're doing your experiments, and I know that that's the case for other instruments. You have variable resolving power, so you can actually switch resolution settings. So you have 3 resolution settings from the minimum to maximum, and you can actually play that, and it becomes actually a really good tool for your different type of experiments. And at the top, the number one reason really why the Exactive Focus is better than QTOF is because of its precise quantitation. And I want to really stress this out because what you get is you get a quantitation performance that of a triple quadruple, and you can do all the other screening and confirmation as well. So you have excellent linearity with automatic gain control at low and high concentration. And I'll actually show you a couple of examples of how good the instrument can quantitate on. So first, let me tell you that there's different ways of doing quantitation on the instrument. So you can quantitate with full scan, you can quantitate with SIM, but you can also quantitate with PRM. And PRM is power reaction monitoring. So we're calling that in analogies with SRM, like triple quadruple. But so it's a little bit different than SRM because obviously we don't have 2 quadruples, we have 2 quads and an Orbitrap at the end. So how the PRM works is that first you do isolation with the quadruple, then you do fragmentation in the HCD cell, and then you do detection with the Orbitrap analyzer. You get your fragment ions and MS spectra, right? And then you quantitate it. You do the quantitation of extracted ion chromatically. So that's how PRM works, very analogous, let's say, to SRM to guide you that way, and with key differences and key benefits there. So PRM combines high selectivity, high sensitivity, high throughput quantitation with confident targeted compound confirmation, right? So you can do all of that as well as confirmation, and on top you get the screening as well. So it's suitable for quantitating tens of hundreds of targeted compounds in complex matrices. You can easily do that, and it's easy and fast to set up. So I want to give you an example of limited detection and dynamic range of Exactive Focus versus a vendor S triple quadruple. So this is actually a compound called Tamiflu. That you buy actually over the doctor prescribed drug. And this is an experiment in plasma, and we have the same settings in terms of the LC settings, the same samples, no different conditions. So same conditions both in Focus and the triple quadruple, where we run this experiment in 3 different scan modes: full scan, SIM or selective ion monitoring, and PRM. And with PRM mode, you see that we got to about 1 ppt limit of detection in the Q-exactive focus with calibration range from 4 ppts to about 1,500 ppbs with a dynamic range of 5 orders. And you can see the calibration curve on the bottom, which is pretty linear. Excuse me. On the vendor S super quadrupole with the in SRM mode, the limit of detection is about 1 ppm, and the calibration range was from 4 ppts to about 150 ppbs, giving you approximately 4 orders of magnitude. So the calibration range was about 10 times less. And if you look at the bottom calibration curve, you see that it becomes really non-linear with the 2 dots circled in red. So we get, in summary, I guess, the Q-exactive focus with PRM mode or in PRM mode has the same limit of detection as a high-end triple quadrupole with linear, with wider linear dynamic range as shown on this slide. Okay, so in conclusion, I wanna kind of point out 2 different aspects of the instrument and summarize it really quickly. On the left-hand side, I kind of pointed out a few things about calibration and maintenance. So you have 5-minute external calibration routine that you do when you wake up in the morning, and you come to the lab and you start running the instrument. You can do that, right? But if you calibrate the instrument in the morning on Monday, it will be fine all the way till Friday. But it's really quick. It's only 5 minutes. There's no internal calibration, mass calibration needed. You can do it, but you don't need it. With external, you're pretty fine to get really good mass accuracy, like I've shown before. Like I said, calibration holds for a week time. There's really very little influence on mass calibration with small temperature variation fluctuations. Of course, I'm talking about just a few degrees, but you don't need to worry about your instrument being calibrated if you open the window or not. The transfer tube is cleaned within 10 minutes. Yeah, you can actually just remove it without breaking the vacuum, and you can put a new one, or you can clean that and put it back there, and so you're ready to go. Inner source cleaning, you do that once or twice a year, and it's about half a day. So typically, I recommend these, the customers to do that cleaning at the end of the day, put it together, and then vent it, and bake it out, pump the system, bake it out overnight, and then you're good to go on the next day. So Q-exactive, so on the right-hand side, I just want to show you how Q-exactive focus is designed for routine work, right? To do your method development, right, or when you're doing your method development, it really takes little to no MS optimization for each analyte. You don't need to worry about your optimizing your transition or your color or your fragmentation energies. You don't need to do anything like that you do on a triple quadrupole instrument. You have high sensitivity and high selectivity. You can do quantitation without limitation on the number of monitored analytes. You can, in addition to your quantitative aspect, you can do confirmation of your analytes using isotope pattern, using the fragments, or exact mass. You can do retrospective data analysis, which means that once you acquire data and it's saved, you can always reprocess. You can always go back and reprocess the same data and look at things, look at compounds that you didn't look before, because there could be something in your data that you never really looked for, but it was there. So you can reprocess it. That's what we mean by retrospective data evaluation. And really is a very affordable, true high-resolution active mass instrument. Okay, so with that, I'd like to thank you for your attention.
And now we'll hear from Kristine Van Natta. Kristine Van Natta is a strategic marketing applications specialist with Thermo Fisher Scientific, working on clinical and toxicological applications for mass spectrometry. She has a degree in chemistry from the University of California at San Diego and a California single subject teaching credential in chemistry. Ms. Van Natta has been working in the analytical chemistry field for over 17 years. Her experience includes extensive method development and work performed under both EPA and FDA GLPs. Prior to joining Thermo Fisher Scientific, she worked for Roche Pharmaceuticals in Palo Alto, California, where she developed and validated GLP-compliant methods on LC-MS systems. I will now turn it over to Ms. Van Natta.
Thank you for the introduction. Good morning, good afternoon, and good evening to our viewers around the world. Today I'm going to discuss some of the diverse forensic applications that can be run on a high-resolution accurate mass Orbitrap-based mass spectrometer. The Thermo Scientific Q-Exactive Focus, a hybrid quadrupole-Orbitrap mass spectrometer, is a highly versatile platform. Some of the applications I am going to discuss include screening, both targeted and unknown, quantitative confirmation, research applications including structural elucidation of unknown compounds, and bulk substance analysis using the DART ion source. I'm going to start with some screening applications. Data acquisition methods for both targeted and unknown screening are quite similar, both involving acquisition of full scan data and then fragmentation data. The primary difference is in the data processing. I'm going to talk about 3 different types of screening methods. The first method starts with the acquisition of full scan positive ion data at a high resolution, followed by all ion fragmentation in the collision cell and acquisition of the resulting MS-MS fragment data at a slightly lower but still high resolution. This is followed by a fast polarity switch and the acquisition of the same types of data in negative ion mode. Using this method, compounds can be identified based on the exact mass, their retention time, and isotopic pattern, all from the full scan MS data, and then by matching MS-MS fragments from the all-ion fragmentation data. This method also allows for complete retrospective data analysis using both the full scan and the fragmentation data. The second screening method is a slight variation on the previous method. The difference is that the all-ion fragmentation data are collected in several precursor mass segments instead of over the full mass range at once. This improves the all-ion fragmentation specificity and still allows for retrospective data analysis. As in the previous method, compounds are identified based on exact mass, retention time, and isotopic pattern from the full scan data and fragments observed in the all-ion fragmentation spectra. A third data acquisition method provides greater specificity. As previously, full scan positive ion data is acquired at high resolution. Here, the full scan is followed by a data-dependent fragmentation scan, or I will say DDMS2. That scan can either be triggered by an inclusion list of specific compounds or by using the most abundant M/Z detected in the full scan. Again, polarity is switched and the same data is collected in negative ionization mode. In this method, compounds are identified based on exact mass, retention time, and isotopic pattern from the full scan. However, instead of just MS-MS fragments, we now have a precursor-specific MS-MS spectra that can be used for confirmation. This is the most specific screening application because the high-resolution MS2 spectrum is a very specific identifier. The trade-off for this specificity is that retrospective data analysis is limited to data extracted from the full scan MS spectra. This data still provides exact mass, retention time, and isotopic pattern, but we don't have MS-MS fragments from the all-ion fragmentation to assist with the identification of unknowns that were not in the targeted list. Here is an example of this last type of method implemented on the Q-Exactive Focus. On the right-hand side, you see the method parameters. We are collecting the full scan MS data at a resolution of 70,000 and the data-dependent MS-MS confirmation data at a resolution of 17.5 thousand. The MS2 spectra were collected in time windows specified in the inclusion list with stepped collision energies to allow fragmentation of chemically diverse compounds and to ensure nice full rich spectra. On the left side, you see the mass spectrum method inclusion list of targeted compounds. This particular list contains over 300 compounds from different classes which we analyzed to test this method. If we had chosen not to use an inclusion list, but trigger the data-dependent MS2 scans based on ion abundance, then we could have also used an exclusion list containing common endogenous matrix compounds that can be added to your method. This list will prevent the acquisition of data from compounds that are irrelevant to your screening and improve your hit count. We developed a screening application for urine analysis using a 20-fold urine dilution for sample preparation and a short 10-minute chromatographic method. Sample preparation was simple dilution with no hydrolysis. A single internal standard, tolbutamide D9, was added. We use this compound because it ionizes well in both positive and negative modes. The column was a Thermo Scientific AccuCore PFP fused core column, and mobile phases were water and methanol, both with 10 millimolar ammonium formate. For automated processing of the data, we use ToxFinder software. It is an intuitive software providing fast data processing and rapid data review. Here you see a schematic of the application workflow. The workflow is as follows. In the first step, you create a database containing a list of target analytes with related identification criteria. The criteria can include information such as the exact mass, retention time, compound-specific fragments, ion ratios, and other identification criteria and limits. Optional is the inclusion of an MS spectral library. Users can either create their own MS2 spectral library or use a library provided by Thermo Scientific. Next, users will create a processing method in which compound identification criteria, peak integration parameters, and report templates are selected. Next, the sample list is created and processed. The data review step is optional. Reports can be printed directly after data processing or after data review. Libraries that can be used in the ToxFinder software include the Thermo Scientific Clinical Research and Forensic Toxicology Library and the Thermo Scientific Food Safety and Environmental Library. The Clinical Research Forensic Library contains over 900 compounds. The Food Safety and Environmental Library contains over 1,600 compounds. Each library entry includes 5 spectra for compound. 3 of the spectra were collected at single collision energies, and 2 spectra were collected using different stepped collision energies. This helps ensure detection of your compounds regardless of the collision energy you choose for your specific method. This is the ToxFinder data review page. From the top left, you see an example of the sample list, and to the right, the data review table for a selected sample. The data review table shows information about the status of each analyte. Here, I sorted based on the status. The green dot in the confirm column indicates that the analyte was detected and identified with all identification parameters specified in the method. In this case, we used exact mass, retention time, isotopic pattern, and the MS2 spectra. On the bottom, we see the results for a specific compound found within this sample, in this case oxycodone. On the left is the ion chromatogram extracted from the full scan data, reconstructed with the mass accuracy specified in the processing method. In the center are the experimental MS spectra acquired for this compound and the corresponding matched library spectra. On the right are the calculated isotopic pattern for oxycodone and the isotopic pattern from the experimental data. Moving on from a targeted, or what we sometimes call known unknown screening, we can now examine what we call untargeted, or sometimes unknown unknown screening. This is the case where you are trying to identify all chromatographic peaks detected during an analysis, including those compounds that you were not specifically looking for. For this data analysis, we will use Thermo Scientific TraceFinder software. It performs a 2-step process to identify unknowns. In step 1 is a targeted search against a local database of compounds. In step 2, for peaks that do not correspond to any in the database, the software will propose a molecular formula based on the mass and isotopic pattern, and then perform a ChemSpider search on that formula. Here is where the importance of the high resolution and accurate mass data really comes into play. With better mass accuracy, the number of possible molecular formulas goes down, thereby decreasing the number of possible unknowns. This is an example of a compound identified in step 1. Lamotrigine in a urine sample was identified from the database with information based on accurate mass, M/Z, and retention time. It was confirmed with the isotopic pattern matching and a library search not shown here. Here is an example of a compound identified by step 2. A molecular formula was proposed based on the accurate mass and isotopic pattern. The ChemSpider database search returned creatinine as a match, which was confirmed by the isotopic pattern matching as well as the accurate mass. For unknown screening, we need a large number of spectral libraries to match those compounds to. In addition to the libraries discussed earlier, I would like to present a new development related to MS2 spectral libraries. Thermo Scientific has established an open access spectra database. The website address is mzcloud.org. This curated database contains multiple MS2 spectra and MS-to-the-N spectral trees collected with multiple collision energies for compound identification and structural elucidation. As of today, the database contains spectra for over 3,000 compounds. It is growing all the time with the addition of data for more compounds collected with the help of collaborators. On the left side of the screen, you see just the first 5 compounds listed in the database. To the right is a spectrum tree for one of the selected compounds, dopamine, and below just one of the MS2 spectra that are stored in the database. Now let's shift from screening applications to one of the many research applications that can be performed with the Exactive Focus mass spectrometer, structural elucidation of a novel compound. We know quite a lot about drugs of abuse around the world. Completely new active pharmacophores are very rare. Often synthetic analogs are created by small modifications to known drugs to thereby avoid detection. Targeted screening won't find them since the molecular weights are different. We can exploit the similar base structures, which will generate similar fragments. It is often possible to identify the chemical class based on similar fragments and from there construct a possible formula for an unknown compound. The process is very similar to finding metabolites of drugs, which we have great tools for. We can rely on the chemical similarity of new designer drugs to help us find it. Since we know other chemical compounds in the class, we can use their fragments to aid in the search. This is a real human urine sample analyzed and provided by a customer using an Exactive Mass Spectrometer with full scan and all-ion fragmentation. Using another software package, Thermo Scientific Mass Frontier software, a FISH, or fragment ion search, was conducted to try and identify an unknown compound based on the similar fragments in the compound class. If we apply this FISH, what do we find? In this case, we found MDPV, methylene dioxypyrovalerone, was identified. Even though our targeted screen missed this compound because it wasn't in our target list, the chemical structure revealed its identity. The common fragments shared with previously known drugs exposed this peak here in the pink trace, and we were able to start focusing on this potentially new drug of abuse. Next, I'm going to discuss quantitative confirmation using the Exactive Focus. There are 3 approaches to quantitation. The first is full scan MS with data-dependent MS-MS. With this method, you quantitate using the extracted ion chromatogram from the precursor mass of interest. The high resolution of the Orbitrap data provides selectivity, and the MS-MS spectra are used for confirmation. This method is useful for samples with less complex backgrounds. One great advantage of this method is that it requires little method development, since all you need to know are the masses of the precursor ions. A second method is targeted selected ion monitoring, followed by data-dependent MS-MS. With this method, only targeted masses are isolated by the quadrupole and analyzed in the Orbitrap detector. This scan is followed by fragmentation scans of the same precursor mass. This enhances performance in more complex matrices by only scanning for compounds of interest and conversely excluding anything that you are not interested in. The most specific method for quantitation is parallel reaction monitoring, or PRM. It is the most similar to traditional triple quadrupole SRM methods. In PRM, there is no full scan data. Instead, only the targeted precursors are isolated and fragmented. The data collected is the resulting high-resolution fragmentation spectra. From this, you pick quantifying and confirming ions for data processing. Here we have an example of the PRM method as applied to the quantitation of 6 opiates in urine. They are morphine, codeine, hydromorphone, hydrocodone, oxymorphone, and oxycodone. Sample preparation was hydrolysis followed by 40-fold urine dilution. In the lower left of this slide, we see the inclusion list with all 6 of the mentioned opiates and their internal standards. On the right side of the slide are the scan parameters for this method, and the spectra were collected with a resolution of 35,000. This is the example of the data you get from a PRM method. On the left is a chromatogram of the fragmentation scans for oxymorphone. On the right is an MS-MS spectra for the compound, highlighting 2 confirming ions and a single quantifying ion picked from the spectra. Here we have reconstructed chromatograms of the quantifying ions in the lowest calibration standards for each of the 6 opiates in this method. As you can see, you get a good number of scans for quantitation across your peak. These are representative calibration curves for codeine and morphine. This method gives excellent linearity and accuracy across the calibration range. On the right are the reconstructed fragment ion chromatograms for a quantifying and confirming ion for each compound. Again, these chromatograms are at the limits of quantitation for these compounds. Overall, this PRM method gave good linearity, limits of quantitation, and precision in both intra- and interassay quality controls. To test the ruggedness and effect of matrices on this method, we analyzed internal standard recovery in many different lots of urine and compared that to the average recovery for the calibrators in artificial urine. As you can see, we have very good recovery, indicating that this is a robust method. And finally, we have some example of opiates in donor urines across the calibration range. Starting from the top left, we have codeine at 12 nanograms per mil, hydrocodone at 60 nanograms per mil, hydromorphone at 37 nanograms per mil. On the bottom, morphine at 217, oxycodone at 4, and oxymorphone at nearly 700 nanograms per milliliter. For my final application, I'm going to show you an example of bulk substance analysis using the DART, or direct analysis in real time, ion source, coupled to the Exactive Focus mass spectrometer. Specifically, we're going to be looking at the analysis of a pharmaceutical pill. We see here the DART source easily coupled to the Exactive Focus mass spectrometer. The quick strip gives you automated analysis of up to 12 samples on the disposable quick strip card. The source is enclosed to minimize sample contamination and operator exposure. Sample preparation for this method couldn't be easier. You simply grind up the pill and deposit a little bit on the quick strip card and tap off the excess. The data acquisition method is also simple, as the DART source only requires 2 parameters, the temperature of the gas and the speed at which the sample moves through the source. The mass spectrometer data acquisition method is similar to what we've seen in previous examples. We acquired high-resolution full-scan positive MS data, followed by a data-dependent MS-MS of compounds and inclusion list. The same types of data are then acquired in negative ionization mode. With this analysis, we've confirmed the compounds expected in this Tylenol number 2 pill. Acetaminophen was detected in both positive and negative ionization modes, and codeine was also found. On the left, you see the total ion chronogram produced as the quick strip moved through the ion source. Full scan and MS-MS scans of acetaminophen in positive and negative modes, and a full scan and MS-MS scans for codeine. On the right, you see the positive and negative acetaminophen spectra and the codeine spectrum. we use ToxFinder software to process the data. Here we have the results for acetaminophen in positive ionization mode. On the left is the reconstructed chronogram for the precursor compound in full scan. In the center is the experimental MS-MS spectrum and the corresponding library matched spectrum. And on the right is the experimental and calculated isotopic patterns. In conclusion, what we have seen here is the versatility of the Exactive Focus hybrid quadrupole-Orbitrap mass spectrometer for forensic applications. It can do everything from screening for known and unknown compounds to quantitative confirmation. You have high confidence in your results thanks to the high resolution and accurate mass data and the ability to use multiple identification and confirmation criteria. It provides high specificity, sensitivity, precision, and robustness. Thank you for attending today's webinar.
Excellent presentations. Thanks for bringing that information to us. Before we get started with all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many as we can. And our first question is, in your opinion, when will routine labs move away from old technology like triple quads to these new HRAM based on Orbitrap for both quantitative and screening on one platform? I believe this one's for Maciej.
Oh, yes, hi. Thanks for the question. Good afternoon and good morning to everyone. So, yeah, that's a very interesting question, and while I travel around the world to many different customers, I see that nowadays customers' demands is much more than just targeted list of compounds. And I've been seeing this for a number of years, where customers are buying the Exactive Orbitrap instrument to do a number of different works in their lab. And there's more and more labs that I see where customers are completely moving away from triple quads, because they want to not only quantitate the targeted compounds, but as well want to identify the unknowns and the non-targeted compounds in their samples. So, you know, it's a tricky question, but when will this happen? But from what we can see is that it will be a few more years, I believe, that you know, this type of routine work will completely go into high-res accurate mass instruments, predominantly on the Orbitrap instrument.
Why is this product used only in forensic labs? Maciej?
Well, it's not. It's not just for forensic lab at all. It's actually for any small molecules, and especially in routine application. So this instrument is not designed for proteomics, absolutely not, but it is designed for routine day-to-day work. It's simplified method editor, it's simplified instrument control software. You can obviously run it like we showed you here and use it for forensic toxicology, but you can also use it for food analysis and environmental, screening water as well, drinking water, sewage water. You can use it for pharmaceutical applications. So it's not just for forensic toxicology, but of course, this was the topic of discussion here today.
And Maciej, this one's for you too. How is the Exactive Focus different from the Exactive Plus?
Yeah, so the Exactive Plus is a really high-end instrument designed predominantly for proteomics application. It has a very different hardware. There are several things that are quite different, like the quadrupole, for example, is a segmented quadrupole as opposed to a hyper quadrupole on the Exactive Focus, which we showed you today. It has a protein mode and enhanced resolution mode, so it's a very different in a way performing instrument that is used, like I said in the beginning, for proteomics applications, and for that it's not really a viable instrument for routine workflows.
This next one, I believe this one's for you, Kristine. How can Thermo's LC-MS use for designer drugs of abuse constant change? I hope I read that right.
Okay. Thank you for that question. I believe we have some grammar issues, but I'll do the best I can. I believe they're asking how can these instruments find the new designer drugs that are constantly changing. And in the screening applications, when you're running the full scan data, you will always get the parent mass. So when you go back in the retrospective data analysis, you can go back and find those new compounds. You can see if it was in your previous samples. And also, as soon as one of these new designer drugs is identified and isolated and we obtain the spectra for it, then that spectra will go into MZ Cloud and you will be able to search on the new designer compounds. If you're searching for these new compounds yourself, you will have to use some of the other software that I showed here, Mass Frontier, and do a little more sleuthing to tease it out yourself.
Okay. And Kristine, here's another one for you. How many compounds can be analyzed in screening applications?
The screening applications can handle a virtually unlimited number of compounds. Since you're not going to be detecting all of your compounds in all of your samples, even if you have a very large inclusion list or a very large database of compounds, the instrument will only be taking data-dependent spectra of the compounds, of the masses that it sees at a high intensity. So because of that, you have a virtually unlimited number of compounds that you can look at. And again, with the full scan data, you will have your complete precursor ion spectra data collected to go back and look at any time.
What sample preparation method and what LC method would you recommend for screening applications? Kristine, I believe this will be for you.
Well, for the screening applications, we recommend a simple and generic sample preparation method. For urine, it would be simple urine dilution. For blood or plasma or something like that, a simple protein precipitation. We have seen posters where people have been using mixed mode or multi-analyte sample preparation type columns for sample preparation. The key in screening, since you have such a wide variety of compounds, you want it to be a very generic sample preparation method so that you don't lose any of your samples during sample preparation. And also for the LC method, we have some nice columns. The columns that I have shown here work very well. And we often go to a neutral pH on the mobile phase to, again, accommodate a wide variety of compounds, polar and nonpolar, acidic and basic. So those are my recommendations.
Okay, and just want to let people know that polls are being shown on the screen. Please respond to them. And we have another question. I think, Kristine, this would be for you. Can you easily swap heavy ionization sources with DART?
Oh, very easily. These sources are very easy to swap out. There's no configuration in the software required, or very minimal. The physical transfer of these sources is just a couple of thumb switches to pull it off and put the new source on. And then in the case of the DART, to also hook up some gas to make sure that's there. And it can be done in literally less than 10 minutes.
Okay. Give me a moment here. I see it may be that more questions are coming in. I'm just going to pause for a quick moment. Okay, well, I believe we will conclude now, and if we did not get to your question today, if you did just send us a question, if we did not get to it, we will follow up with you via email. I would like to thank our speakers today, Dr. Maciej Bromirski and Kristine Van Natta, as well as our sponsor, Thermo Fisher Scientific, for making it possible to bring this presentation to you. Today's webcast will be available for on-demand viewing through April 1st, 2016. You will receive an email from us alerting you when it's available on demand and posted on labroots.com. You are welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. See you next time.